I guess I'll go ahead and introduce you. So, Maddie is a senior biochemistry major with a psychology minor, and after graduation, she's going to work in the medical field for about a year before going to med school. And over this past summer, she did an REU at UT, so she's going to tell us about that today, and then is going to stick around for some questions afterwards if you have any. So thanks, Maddie, for giving us your time, and I'll hand it over to you.
Thank you, Julia. It's an honor to be introduced by you. Okay, I'm gonna share my screen, and if someone could give me a thumbs up that it's working. I'm super excited to be here today. I have friends and mentors from near and far, and also some new faces from MC, so it's super great to see all of you, or at least see your names. And today I'm gonna talk about the effects of the regulatory mechanism on the Lux gene cassette, testing efficiency of bioluminescence using the NF-kappa B response element. And I'm also going to talk about how to apply for an REU, especially during a pandemic, and what it looks like to complete an REU as well. And if you have questions along the way, you can use the chat box, but I probably won't see it. So if you'll just hold off until the end, I'll also leave plenty of time for questions. So the first thing I'm going to talk about is planning your applications. So REUs roll over pretty quick, like some of the applications are due very fast at the beginning of the new year. So I suggest you start planning over Christmas break the year before you want to complete one in the summer. And the first thing that I did to keep myself organized was I made an Excel. And on the next slide, I'm going to show you kind of like what I put in my Excel and what I was thinking about when I was thinking about each program. And the second thing that I did was I wrote an outline cover letter. And then I would change it for each specific program, but I kept the same outline story for each, and then I would tailor it from there, because that saves you a little bit of time, so you don't have to write an entire letter for every single program, because you're going to want to apply for multiple. And then the last thing is ask for your recommendation letters early. And I'm also going to show you what my emails to those professors look like and what I gave them so that they could write a recommendation letter that was tailored to me and to the program. So these are things to consider when picking a program, and these are things that I put in my Excel sheet. I was always looking at program type. So is it biology, chemistry, biochemistry, physics? And also from the program type, you're probably going to want to tailor your references to whatever that program is. So when I applied for physics programs, I always asked Dr. Guerno to be my recommender because that made the most sense. The second thing that you're going to think about is pay or benefits, and that also goes along with the location of your program. Normally housing is included, but that's always something that you want to look into because, for instance, during the pandemic, my housing was not included. Therefore, I had to figure out rent and like what my budget for the summer looked like because of that. And also, just because the pay might seem higher in some areas, it might also just be that because of the location, the cost of living is a little bit higher. So those are just things that you want to be aware of as you're applying. The next thing is a deadline. Like I said, some of the deadlines roll over super fast and others will be as late as April. So you might want to triage your approach to which ones you're applying to according to the press of the deadlines. And then one of the questions that I always ask the professors or the PIs in charge of this was, can you do your thesis with this? This is such a great atmosphere to do your thesis in because you can commit so much time to it. And also because you're going to be around a lot of other people who are motivated and working just like you. And it's like college. When other people around you are doing really great things, you're also motivated to do those things as well. And the last thing is class requirements. Depending on the lab that you're going into or the research that you're going to be doing, there might be some class requirements that you have to have so that you'll know what to do while you're in the REU. This is quite literally a snapshot of what I sent my professors. Mind you, for reference, this person had already said that they would be my recommender. So in this letter, I'm not exactly asking them to be my recommender. I'm more giving them the information that they might need to write my recommendation letter. So I would say, dear so-and-so, and then I would just explain what I was doing. And I pulled this little chart straight from my Excel sheet. And that is truly the easiest way to do it, to get the professors the information that they need and also to keep your information in order. So I would put the program, the due date, and the website for the professors, just in case they wanted to go look up the website and see what that program was looking for, or just if they were curious, so that they had easy access to that information. And then I would also attach my generalized personal statement and my resume, because again, you want to make it as easy as possible for these professors to write you great recommendation letters that are tailored to you. Upon acceptance, be excited. Like, it's an awesome thing that you got into an REU, and you can definitely email and say thank you and that you're excited to the PIs and to whoever accepted you. I did, and they were really appreciative of that, because apparently sometimes people just don't, because they think it's unprofessional. But I definitely think that you can be professional excited at the same time. And yeah, that's just, you should pat yourself on the back for that if you get in. It's a really great feeling, and it's a really great thing. And then the next thing that you might want to do, just like when you move to college, when you're doing an REU in a typical year, and I imagine that they're going to get back to somewhat normal this year, you're likely going to have suite mates on campus. And so you might want to reach out to them and ask them, who's bringing the dishes, who's bringing the laundry detergent, those things, so that you can more efficiently work with the people that you're going to be living with. This is a list of tips for success while doing an REU that I made. The first thing is definitely stay organized. So in the beginning, you're going to tell yourself that you're going to remember things. And then by week 2, you're not going to remember what you did a week ago. And so I definitely got that little life check in the second week. And I then started keeping an online lab notebook. I know we do paper here at MC for various reasons, one of them being grading. But when you're working in a lab, you're going to be communicating so frequently with your supervisor that it's almost easier to keep an online notebook, so you can just send it back and forth between the two of you and make little tweaks virtually. So that's what I did. And then the second thing is, when you're assigned to a project, make the effort to research what you're doing independently before going and asking all of the questions to your supervisor. Obviously, they don't expect you to come in being an expert, but they do want to see that you are putting the effort in to work independently and to understand these things, and not just coming up to them and saying like, oh, I need you to explain the whole thing. Like, that's not what you want to do. You want to go and try to understand the fundamentals of what you're doing, and then from there, go ask them for clarifying questions, and they'll help you with that. But it's just a really great skill to have, to take it upon yourself to research these things first. The third thing is, if you are using this experience to write your thesis, work ahead. It's going to make it so much easier on you. And I suggest writing, like, whenever you learn a new skill or a new technique or something about the project, go ahead and write your introduction paragraph on that piece so that it's fresh, and it also helps you learn it even better. And then your thesis will be done by the end of the summer, which is awesome. I wasn't quite to that point, but I definitely did try to work ahead. And so this is just me also learning from my mistakes, telling you it would be even better if you did work really far ahead. And then the next thing is to always be making those professional connections with the people that you're working with. Those are just great connections to have. And even since leaving the lab at the end of the summer, I've talked to the PI and the other researchers in the lab since then. So just keeping in contact with those connections is great. And the last thing, and this is really important, you need to maintain a fair work-life balance. You're there to work, and so you should definitely put in a lot of work. I know that I did. I was there on the weekends all the time, and that's a really great thing, but you don't want to burn out, especially if this is what you're looking to do for your career. You kind of need to learn how to make that work-life balance work early, so that going forward, you're going to be really ready and really fueled to do some really awesome work. Now I'm going to transition over to talking about what I did my research in the summer on, and also what I wrote my thesis on, which is, again, the effects of the regulatory mechanism of the Lux gene cassette testing efficiency of bioluminescence using the NF-kappa B response element. I did this at the Center for Environmental Biotechnology at the University of Tennessee at Knoxville. And then my thesis advisor here at Maryville was Dr. Duncan. Oh, I'm going to go back for 1 second. The reason why I picked the photo of the fireflies here is because the Lux cassette is roughly based off of the Luck cassette, which actually comes from fireflies. So basically what this project is doing is taking something really beautiful from nature and tailoring it to what we want to do with it, which I think is the coolest part of this whole thing. The first thing that I want to talk about, though, is that's a lot of words, and we're going to go over a lot of techniques, and so it's going to seem really complicated, but I wanted to tell you the why of why we're doing this research so that this doesn't get lost in translation. The why. So some issues that we have right now in the medical field are that there are some pretty common reasons why people don't want to seek medical care. The first being unfavorable views of the medical field, and the reason for their unfavorable views often comes from the pain associated with testing and the time that it takes to get that testing. So for instance, a lot of people won't get their yearly checkups on things that they're supposed to, just because it might be uncomfortable and it might bring them bad emotions. And then from that, this problem also stems from that. People will become accustomed to their symptoms as they're normal, and then they're not going to go to the doctor because they've lived with it for so long that they just don't see it as an issue anymore. But that stems from the original problem of the pain and just the discomfort of going to the doctor. And the third thing is traditional barriers that we think of, such as the high cost of medical care, low access to resources. For instance, if they don't have insurance, that's going to cost them even more. And then also time constraints, especially if you live in rural areas. It's very hard to get to the medical care or the medical facility that you might need to do your testing. So those are things that we're trying to work on. Therefore, the goals in research are to create more non-invasive and less painful methods for testing so that people will not necessarily avoid going to get testing for things, and they might actually get the medical help that they need. And then these testing methods also need to be relatively inexpensive and relatively efficient. Now, that was some big umbrella stuff. I'm going to talk about the very small umbrella of what I was working with. So this is the background about the research that I was doing. So the bacterial luciferase gene cassette is composed of 5 essential genes, LUX C, D, A, B, and E, and they're in that order because that's the order that they are on the DNA. And these 5 genes allow for the bacterial species to be independently capable of producing light. However, there is a 6th gene called FRP, which is not necessary for the production of light, but it has been seen to increase bioluminescence. So you're almost always going to see that in my samples, because again, our goal is to increase the bioluminescence so that we can strengthen our testing methods. And in figure 1 here, what it means by being independently capable of producing light, it means that the species can gather the cofactors that it needs to produce light on its own. And in figure 1, it shows you that the cofactors are mystric acid, FMN, and molecular oxygen. And also the interesting thing about this cassette is that it doesn't all have to be in the same portion. Like you can have 2 portions of it in the cell. And if you have every component, no matter how many pieces it's split up into, it will still produce light. The second thing that I want to talk about is nuclear factor kappa B response element, or NF kappa B, which contains transcription factors that have been tied to inflammation, immunity, cell proliferation, differentiation, and cell survival. And I hope some of these words are bringing up some things like cancer and immune disease, because that's exactly right. That's exactly why we picked to put the LUX cassette under the control of NF kappa B, because in this research specifically, we're looking at developing cancer-like models for the cell. So that's why we put it under the control of NF kappa B. To continue with background, the LUX reporter-based system is a real-time and non-invasive way of measuring the NF kappa B activation. And again, going off of the cancer model here, we're using a chemotherapy drug that's been known to cause NF kappa B activation called doxorubicin. But as you can see in figure 2 here, this is the organic line structure of doxorubicin. But note that the molecular weight is massive. For a drug, it's 543.5. And as you can see, it also has lots of polar groups around it. So this drug being so large and having those polar groups has a lot of room to be toxic. So along with testing the different regulatory mechanisms, we're also testing the effective dose and the toxicity of doxorubicin as well. Okay, so this slide is a little wordy, bear with me, because on the next slide, I'm going to show you pictures of all of the things that I'm talking about. So the first thing that you have to do when you're working with DNA is you actually have to collect the DNA. So when I was extracting DNA, most of the time I extracted it from E. coli because E. coli is really easy to grow and it's very fast growing. I extracted it using the Zymo Pure 2 Mini Prep and the Zippy Plasmid Mini Prep kits. And I would use the Mini Prep kit when I was testing something new and I didn't want to waste a lot of reagents until I knew that it was what I wanted. And then I would use the Mini Prep kits when I needed more DNA to do the transfection, for instance. The second thing is that I designed the target plasmids that we wanted using the APE software. And if you've had genetics or biochem, you know what I'm talking about. If you haven't, that's probably something that you get to look forward to. And then after you've designed it into the APE software, you can see if it's possible to do restriction, digest, and ligation with 2 different pieces of DNA to create that target plasmid. And if it's possible, we use that method because we already had the restriction digest enzymes in the lab. However, if that's not possible, that's when I would have to go and make Gibson assembly primers and then do Gibson assembly and PCR to get the DNA that we wanted. And I have a lot of respect for the professors that make the Gibson assembly primers because it was far more complicated than I expected, and I definitely messed it up the first couple of times. So the next thing that I would do is take the component and single gene regulators, which you'll see what I'm talking about in a minute, and transform them into 5-alpha E. coli cells. However, with the full cassette, I transform them into the 10-beta E. coli cells. The reason for that being they're relatively very, very similar. However, the 10-beta cells are a little bit more competent at picking up large pieces of DNA, and the full cassette was larger than the component or single gene regulators. So that was why the choice was made there. And then from there, the new plasmid DNA was extracted from the E. coli, screened. Again, when you're doing research, you always want to make sure to screen your work and make it fairly easy to go back one step as opposed to getting all the way to the end, realizing that you've messed up and go all the way back to the start. I screened these things all the time to make sure that I knew what it was. When you're working with DNA, you can't look in the tube and see if you got it right. So it's really important to screen it often. And the last thing is once the DNA was cleared and screened and all that was good, the DNA was transfected into either U87 glioblastoma cells or HEK embryonic kidney cells. And the reason for those choices being, again, we're trying to make a cancer model. So glioblastoma is a type of brain cancer. That's why we made that choice. And then the embryonic kidney cells. The kidneys are an organ that you often look at in the body in association to looking at the toxicity of drugs. So that's why we made that choice, was to be able to make a model of kind of what the human body might look like. Okay, now for the pictures. This is going to help a lot more. So figure 3 is a figure that I made to teach myself this process because it confused me a little bit as well. So you use restriction digest enzymes, such as the ones that you see at the top, to cut 2 pieces of DNA. And it basically cuts and creates either 5' and or 3' overhangs, which are called sticky ends. And then when you put in DNA ligase, it's going to ligase the complementary pieces of DNA together. And it gives you the target plasmid that you were trying to create, if this works. But restriction digest enzymes only work on specific sequences. So sometimes if I didn't have the specific sequence where I needed it, then we would have to go in and make those Gibson assembly primers. Figure 4 is showing you the final target plasmids that I was hoping to create. Over the summer, I did make all of them. However, I was in the process of screening the last 3 when I had to leave because my REU was complete and I had to come back to school. So since then, those have been screened and the assays have been run on them, but that data is not in this presentation because I didn't finish it before I left. And that's totally okay. When you're doing really large projects like this, you're not going to be the end all be all, and that's totally fine. But just know also that not all of your data that you produce is going to end up in the presentation or in the thesis. For instance, these are just the targets that I've listed for basically my thesis. However, over the summer, I probably made upwards of 30 different target plasmids. They're just not in this paper because these are the ones that we decided to test for this, if that makes sense. And then the last figure, figure 5, is showing you the 4-day process of transfection. I'm using the U87 cells as the example. However, this could be any human cell line, but for us, we use the U87 cells. So on day 1, you're going to plate these cells into a 96-well plate. And on day 2, you're going to transfect the plasmid DNA into the U87 cells. And one thing that I found over the summer is working with human immortalized cell lines. They are very, very picky, and it's very easy to kill them. So when you're doing transfection, you want to work very quickly, and you want to make the cells uncomfortable enough that they will take up your foreign DNA, but you don't want to make them so uncomfortable that they die on you. So you have to work quickly, and you have to work extremely sterilely when working with these types of cells. On day 3, the U87 cells were transfected, or the U87 transfected cells. So once the transfection is complete, you then treat them with your 4 different levels of doxorubicin. And you also always include your transfection and treatment controls. If your controls are bad, your experiment is bad. So then you'll have to redo it, but you always want to put them in there so that you can check yourself. The last day, day 4, is when you obtain the results from the plate reader. And we wanted to gather a lot of data, again, about the toxicity of doxorubicin, but we always wanted to also see like when it started working and when it kind of tapered off. So we gathered continuous data over a period of about 24 hours to see when the drug was at its peak and when it started to cease to work anymore. Okay, these results here. So these are results that I wouldn't exactly put in my thesis. I would probably put them in the appendix, but not in the results section, because this is more of me checking myself. But I wanted to show you all because it pertains to a lot of the things that you might do in your classes, and also just gives you more background on what I was doing over the summer. So using the APE software and Excel, I would make little tables like this where I would take my target DNA that I thought I had created and I would digest them with 2 enzymes that would tell me if I got the target plasmid or not. So I would specifically pick those, and then I would get the expected size from the APE software, and I would put it in a table like this so it was easily accessible to me and my supervisor. And then from there, on figure 6, I would make a 1% gel, agarose gel, and I would load my gel with a ladder so that you can tell the size of the DNA, and also with my different clones that I thought that I had made, and I would run it for about an hour. It depended on the voltage that I wanted to run it at and how accurate I wanted to be. But this is also just a very crude method of checking yourself. So this isn't like I'm absolutely quantifying the size of DNA. This is more of a rough estimate type thing. But I really liked this gel because it has a couple of things that went wrong and a couple of things that went right. So I can kind of show you. So on clones A1 through 4, you can see that the digestion happened as I expected it to, as there's a band here around 8,000 and there is one here around 750. However, in clone A5, I really don't know what happened or what pieces of DNA it picked up. So this is one that we would then discard because we don't want to transfect it and waste our time, essentially. And then all the C clones looked great, and the FRP clones here, 1 through 4, look great again. But on FRP clone 5, what appears to have happened is that during the original restriction, digest, and ligation, the DNA likely did not do that properly because it does not look like it took up the additional pieces of DNA that I wanted because it didn't digest at all. So just things to note, and I did a lot of this over the summer. And then once I saw which ones were good, I would check the purity and the concentration, and then we would transfect the ones that had the best purity and concentration from there. So another example of my results, and this was one of the earliest samples that we did because we were also testing the ratio of basically the experimental piece that we made and all of the other components of the cassette. So obviously if I'm testing a single gene regulator, let's say Lux A, I have to supplement it with Lux B, C, D, E, and FRP, or it's not going to be able of independently producing light. So in example, or figure 7, you'll see that we used a 1-to-1 ratio of the experimental and then the additional parts of the cassette that it needed. And in figure 8, we used a 1-to-5 ratio there. And this was to test and see which one was going to give us more bioluminescence for the rest of the samples. And as you can see, they kind of look the same, but this is when it's really important to look at your axes, because if you look at the axes for this one, it only goes up to about 3.7. However, if you look over here, this one goes up to 20. So obviously figure 8, the 1-to-5 ratio, produced a lot more bioluminescence. And that is the ratio that we then went with because that produced the best results. This next slide is me condensing the data that we got for these target plasmids. So again, this is the sample that we just looked at on the previous slide with when we were testing the ratio. From then on, we used a 1-to-5 ratio, as I said. And I was also noting the effective dose of doxorubicin for each sample. And it varies a little, but the big thing that we noticed is anything over 5 microliters started to be toxic to the cell. And that was really what we were looking for, was the toxicity level. And then I also noted the light or luminescence produced. And it's cool to note that as we were going through this research, we also optimized a version of the cassette and we named it JCAT at the end here. So it makes more sense that the JCAT versions of the cassette produce more light. And I'm also going to show you this one in the next slide to show you kind of our best example from the summer. So here you have the NF-kappa B FRP JCAT testing, and you can see the different levels of doxorubicin and my control here. And I always made a bar graph to go along with it because I felt like it was easier to see which level of doxorubicin produced the most light from there. So here it is about 1 micromolar. And this one surprisingly handled the 5 micromolar quite a bit better than the other ones, because normally we would see that at 5 micromolar and above, the bioluminescence would be very low because it was killing the cells at that point. But this one handled it okay. And it also, these results make sense because, like I said, the Lux, CD, A, B, and E are essential for the production of bioluminescence. However, FRP is not, but it does enhance bioluminescence. So this made a lot of sense to me when the data came back this way. And that's another good way to check yourself. Just ask yourself the fundamentals. Does this make sense for what I'm working with? And this result absolutely does. So to summarize, and I'm super excited about this bit, we saw that the effective dose of doxorubicin was found between 100 nanomolar and 1 micromolar, and anything above 5 micromolar was too much. And we also, this is a super exciting part, especially as an undergrad, submitted for publication in the journal Biotechniques, and that was about 2 or 3 weeks ago. So I'm not too sure the turnaround on stuff like that, but it shouldn't be too much longer before we hear if we got in or not. And so just as an undergrad, it's an amazing experience to be a part of something like this and potentially be published. And then for future studies, what they're always trying to do is further optimize the cassette to produce more light, to maybe alter the wavelength, to maybe switch the model up a bit to get better models of humans and what we're trying to look at. So this is an ongoing research project. As I said, I was just a very small piece of it, but it's very exciting to be a small piece and to also continue to see what they're doing now. Acknowledgements. I definitely want to thank the University of Tennessee at Knoxville, especially the PI Dr. Rip and Ting Ting, and the National Institute of Environmental Health Sciences for allowing me to participate in the Summer Undergraduate Research Experience in Environmental Health Sciences. One note about this, when I was notified that I got in, about 3 to 4 days later, I was also notified that the program was canceled because of COVID and because of the uncertainty at the time. However, I took a big jump and I basically emailed Dr. Rip, the PI, and I said, is there any way at all that I could possibly do this? If I find housing for myself, if I find a way to get to UT for myself, things of that nature. Because going into my senior year, I knew that this was my last opportunity, and I knew that it was really something that I wanted to do. So I just thought, why not ask? The worst thing that can happen is that they can say no, and that's okay. But I did ask, and he asked NIH, and NIH said, yes, go ahead with the project. And so I ended up being the only one in the lab. And it was a little bit lonely at times in terms of not having students, but still overall a great experience. And I also got to live with my friends for the summer. So just a great thing that I wouldn't have gotten to do if I hadn't have taken that little risk and asked him that. And then I also want to thank Dr. Brigatti and Dr. Guerno for being my recommenders for the REU, and Dr. Duncan for being my recommender and my thesis advisor and my academic advisor. So thank you. And also thank you to all of the MC faculty, because I wouldn't know how to do any of this if I hadn't taken the classes and learned through the labs. So I suggest to all of you watching, if you're wanting to do an REU, especially take the lab portion of the class serious. And to do that, you also have to take the regular portion of the class very seriously. But when you're doing this, try to understand the fundamentals of what's happening in the lab, because you might forget the details, but if you remember the fundamentals, you're never going to be lost in terms of what you're doing. This is my works cited page. It's very long. And then now, if you have any questions, feel free to do the little raise your hand thing or comment in the chat. And also let me know if you want me to go back to a slide if I need to answer questions about the data at all. So, questions?
I have a question.
Hello. What is your question?
When and how did you decide that applying for an REU was right for you?
Great question. So I was very interested in medicine, but at the time I wasn't sure if I was interested in the patient care side or the research or both. And I was just very interested in lab. Like lab's always been very interesting to me, and I needed a summer job, and I wanted to get that experience and see kind of where I wanted to go from there in life. So that's when I decided that I wanted to apply. To be honest with you, I felt like I started my applications a little bit late in the game. So going forward, what I would suggest for you is start looking over Thanksgiving and Christmas break, and it's going to make it easier for you to narrow down the ones that you really want to apply to. Ooh,
Thank you.
Awesome. I see some questions in the chat. What exactly is environmental biotechnology, and how could you get involved or learn more about this? Okay, so environmental biotechnology, it's basically a very wide term, and I think it can mean a lot of things. But the way that it was kind of used at UT, from my perspective, was it pertained to anything. It was almost like the field where you can bring a lot of fields together. So you can bring physics into it, you can bring biology, chemistry, and it's kind of those projects where you're investigating what's happening in nature or with natural things using a lot of different fields. So I hope that helps some. That's probably not a whole encompassing answer to your question, but hope that helps a little. And then Dr. Gibson, what program did you guys use for electronic lab notebook? We just used Google Drive, and that was really great for us. I've also used Microsoft Teams with my thesis, and that works really great as well. I don't know if I prefer one over the other. I think I prefer Google Drive for if you're working with like a PI or in a lab, because then you can both edit it, but both worked really well for me. So thank you for your question, Dr. Gibson. Any other questions? Matthew?
So my question, I don't know if I just got a little bogged down in the details, but the reason you're researching into the gene for bioluminescence is so you can apply it to certain genes, like for instance, to find like cancer or something.
Yeah, exactly. And you can actually use the bioluminescence to test like a large variety of things. For instance, when I was applying, I was really interested in how they were using bioluminescence to test for water quality. So that's one way that you can use it. That also, for Samantha, that's another way that you can investigate things in terms of environmental biotechnology. But to go into your question a little deeper, Matthew, we were trying to use the bioluminescence as a tool for testing for cancers, and also a model for the human body in terms of, can we try this drug on a person without actually trying it on them to see if it's going to work for their cancer, to kind of prevent pain on their side, if that makes sense. So basically making a human model and using the bioluminescence as the testing factor for that, and testing the drug on their type of cancer to see if it works before you give it to that patient.
That makes sense.
Yeah, and this is also, it's kind of hard to see it because this is kind of not the infancy of the project, but it's definitely more in the beginning. So you're not seeing like an apparatus for testing, but this is just the fundamentals of trying different things and seeing if this will work for testing humans and things like that. So great question. Are there any other questions? And please let me know if I need to clarify things. I know this is complicated.
So is the doxorubicin a drug that's currently being used to treat cancer, or is it kind of one that's in the works?
Good question, Shelby. It is currently being used, I believe. I believe when I checked it was being used. But it has a ton of side effects right now, so it's not the most ideal drug for treating cancers. And as far as I know, it's not commonly used. But the reason why we used it was primarily because we knew that it worked on the NF-kappa B response element. So we knew that it was going to trigger what we needed it to. And it was also kind of complicated for me to understand this bit as well, because you're thinking we want to stop the cancer, right? Like, why do we want to activate this response element? But also, this just has to go back to immunity and the way that the body processes like toxic cells. So basically, from my understanding, you are using the drug to induce the cells to grow a lot so that the body can then recognize it as dangerous and attack the cells from there. Does that make sense?
Yeah, but it's scary.
It is. I mean, cancer is a very scary thing, and our approaches, they're rapidly improving, but it's scary. And yeah, that was a very complicated thing for me to understand too. But good question. And Dr. Gibson also said it is being used. It's kind of an old drug, so since then they've probably produced much better drugs with less toxicity and things like that.
Hi, hello. Great presentation. I just wanted to ask, could you do anything to make the drug more selective for the target cells? Did you look into that?
I'm sure you could. That wasn't exactly my bit of the research, because again, the primary thing for us was to activate the NF-kappa B response element. But like Dr. Gibson said in the chat, I'm sure that since then they've made the drug more selective, and they probably use more of those selective drugs for the cancers that they're treating today. But good question. That's more like organic lab stuff.
Pharmacology, thanks.
No, you're good. Also, Carolina joined us all the way from Poland, and I haven't seen her in years, so I was super excited to see her. Any other questions?
You may have mentioned this in your presentation, but how did you get the plasmid DNA, or no, how did you aggravate the human DNA to get it to accept the plasmid DNA?
Great question. I did not go deep into that in my presentation just for the sake of time, but what you're doing is you're putting it through different solutions and different conditions just to make it uncomfortable enough to take up the DNA. I don't remember the different solutions that you're putting it with like off the top of my head, but you basically treat it with different solutions of things for a very short amount of time while you're doing the transfection, and then you make it very happy again. And the process of working with human cells is amazing. It's something that we don't do at Maryville because it requires like daily attention and daily maintenance. But it was really cool to watch, and the cells require a lot of very specific conditions. So again, you're just using different solutions during the transfection and then making the cells happy again and allowing them to grow from there.
If no one else has any pertinent questions about the study, I'm a freshman, so I haven't read or done a lot of made a lot of biology figures or anything like that. I think it was figure 7 and 8, like the bar graphs, are the top of the actual bars or the points on the other graphs. Are those the medians or the averages of like the range, like represented by like the thin black lines?
I'm glad that you noticed that. I definitely should have mentioned it. When you do it so much, you kind of forget to mention it, but that's a great point. Those are the standard error bars. These long things right here, that's what you're talking about?
Right, yeah.
Yeah, those are your standard error bars. So basically how high or low it's a calculation of how high or low you might be off from that sample. And for instance, I have standard error bars because I took an average for every hour.
All right.
Mm-hmm.
Okay, thank you.
And same thing on the bar graph here. Those are still the standard error bars.
Right.
Good question. And also, if you want a tip for your lab reports in the future, you always want to put it at the bottom here in your figure legend. That's your standard error. So.
Good point.
Anybody else? I know we're getting close to time.
Maddie, can you hear me?
I can.
So much good information, such a lovely presentation, and really great advice to students. So we really appreciate you taking the time. Glad to welcome folks from your other circles in today. And unless there are other questions, I think we can probably wrap it up.
Awesome. Thank you guys so much
All
for
right.
having me.
Thanks so much.